Okay, today we are on the last section of chapter 9, section 9.3. We're still talking about energy, but today we're not going to talk about energy in plants. We're going to talk about cellular respiration. We're going to talk about how that process works. We're not talking about photosynthesis, we're talking about cellular respiration. So let's get into this section. First thing we're going to ask ourselves is, what is cellular respiration? You do not have this slide on your handout, but that's fine. You could still follow along. Second point we're going to look at is the 3 stages of cellular respiration. Then we are going to look at fermentation. You guys ready? Yes. Let's move into it then. All right, cellular respiration. What is it? It's a process of taking oxygen. We're taking glucose, and we're turning it into ATP, energy. Do we need energy? Yes. Do those guys on the screen need energy? Yes. Yes, they're playing football. He just did the pass, the ill pass, and now he's running to the goal. Yeah. Okay, let's see if he makes it. All right, so he needs energy in order to do what he just did. You guys need energy to be here. If you're paying attention in class, do you need energy? Yes. Yes. If you're sleeping in class, do you need energy? Yes. Yes, you need energy for everything. So we're going to talk about how we get this energy. Where do we get glucose from? Sugar. Okay, yes. When we have to take it in, right? We can't, like plants, get energy from the sun. We need to eat, and then we can break down the glucose. We can break down the food that we take in so that we can make ATP, which is going to be the energy source, the energy currency of the body. Do you think cellular respiration is an efficient process? Yeah. I think it is too. Let's look at how efficient it is. What do we have there? You might know what it is. What is it? It's an engine. All right, that's an engine. A gasoline engine. A gasoline engine might be as efficient as 25%, meaning that 75% of that energy is given off as heat. Let's say we have a nice, powerful car. Give me the name of a powerful car. Mustang GT?
Chevelle.
A Chevelle.
1970.
What?
Chevrolet Chevelle.
Uh-huh. 1970? Sweet. All right. So we got a Chevelle 1970. Let's say you're in that car. Let's say you're drag racing. Okay, can we do that? All right, we're drag racing, but we're doing this for a while. We're going over and over and over again, and then we stop the car, and we pop the hood, we take our hand, and we put it on the engine.
You'll probably burn it off.
You'll probably burn what off? Your hand, right? It's going to hurt. Yeah, you're going to burn the engine off, I know. All right, it's going to burn because 75% of the energy that's produced is being given off as heat. Okay, so a lot is lost as heat. Let's look at cellular respiration. Cellular respiration is approximately 40% efficient, with 60% of the energy going to heat. So it's much more efficient than your lovely 1970 Chevelle. 60% of it is given off as heat. Do we want that given off as heat? Is that a good thing? Yeah. Why? Okay, so we want the body to be warm. We live in Michigan. We don't want the body to freeze or anything like that. So it's a good thing that some of it is given off as heat. What is cellular respiration? Cellular respiration, first of all, it happens in 3 phases. And these 3 phases I want you to be very familiar with. Number 1 would be glycolysis. So write that in there. Number 2 would be the citric acid cycle. Another name for the citric acid cycle would be the Krebs cycle. No, we're not talking about the vitamins that you get from citric acid. We're talking about from oranges or anything like that. We're talking about citric acid cycle. Another name would be the Krebs cycle. Why do you think it's called the Krebs cycle?
It's named after a guy with the last name Krebs.
That is exactly what it is. It's named after a guy with the last name Krebs. I've always wanted to describe a process and call it like the Samuel process. I think that'd be awesome. All right, so citric acid cycle. And then we have the electron transport chain. If you are very lazy, you can call it the ETC. How many lazy people we got here? All right, so you can call it the ETC. If you call it the ETC on the test, that's fine, unless I specifically ask you for the name spelled out. And if I do that, then I want you to say electron transport chain. So the first phase is what? Glycolysis. Say that together. What's the first phase? Glycolysis. Second phase would be citric acid cycle, and or you can call that the Krebs cycle. And the last phase would be electron transport chain. Okay, so we have maybe two lazy people calling it the ETC. That's okay. We can survive with that. All right, so the big picture. First, we're starting with glycolysis. The products from glycolysis then go to the citric acid cycle, or the Krebs cycle. And the products from those, some of the products from those go to the electron transport chain, or the ETC. All right? Let's break it down one step at a time. Glycolysis. We're going to talk about some details. I'm going to tell you what the important details are for you to learn at this point. First thing you want to know is that it's a series of chemical reactions in the cytoplasm. What is the cytoplasm? It's in the cell, yes, okay. It's the fluid inside the cell. All right, good. So we're in the cytoplasm of the cell, and we're breaking down glucose and ending up with 2 molecules of pyruvic acid. So we're starting with glucose, and we're ending with pyruvic acid. What are we ending with?
Pyruvic acid.
You guys are on point. 4 ATP molecules are produced in this process. Okay, so 4 molecules of adenosine triphosphate produced in this process. However, the process uses 2 molecules of ATP to get started. So if we're making 4, but we're using 2, what's the net gain? 2. So we have a net gain of 2 ATP molecules, and we're also producing 2 molecules of NADH. Now, here's something I don't want you to get confused with. When we were talking with plants, it wasn't NADH, it was what?
pH.
NADPH, correct. It was NADPH. So when we're dealing with plants and photosynthesis, it's NADPH. When we're talking about cellular respiration, we are talking about NADH. Okay? So how many molecules of ATP are we producing?
4.
Overall?
2.
No, in total? 4. How many are we using? 2. So the net gain would be 2 ATP. Makes sense. All right, sweet. Let's go with looking at the process. Here we have glucose. And you can see it's showing that we have 6. What? Anybody know what those little circles are? Okay, we're basic. I heard it. Carbon. Okay, it's showing that we have 6 carbons. 6. It's C6H12O6. So those represent the 6 carbons. You don't have to know all those details right now. But we're starting with glucose, and here we're using 2 ATPs. 2 molecules of ATPs, and we're making 2 PGAL. You don't have to know what that is, but it's 2 phosphoglyceraldehydes. And from there, we take that and we produce 4 molecules of ATP. We also produce 2 molecules of NADH, and the end product would be 2 molecules of pyruvic acid. We're starting with glucose. We're ending with 2 molecules of pyruvic acid. Those are important details that I want you to remember. Are we good so far?
Yes.
Are we good so far?
Yeah.
All right, good. Starting with glucose, ending with 2 molecules of pyruvic acid. We're using how many molecules of ATP? 2. We're making how many molecules of ATP?
4.
So the net gain would be 2. Sweet. And we're also making some NADH. So notice here, NADH, when it goes from NAD plus to NADH, it's picking up an electron. That's an important thing to remember, and we're going to see why in a little while. Okay, so from glycolysis, what do we go to next? citric acid cycle. But before we get there, we got to do something else. There's an intermediate stage. And in this intermediate stage, following glycolysis, pyruvic acid moves into the mitochondria. And in the mitochondria, it undergoes a series of chemical reactions that causes it to lose one CO2 molecule. So it's losing carbon dioxide. Where does that carbon dioxide go? When we make carbon dioxide, where does it go?
Into the plants.
Okay, so it goes to plants, and it's used in the light-independent reaction. That is correct. It's going to plants. Okay, but here's the important part. There's the intermediate product combines with coenzyme A to make acetyl-CoA. Now, for me to remember this, I have... Oh, you remember. You remember. All right, sweet. So, this is how I remember it. I like to put rhythm to some of the things that I do. Is that okay?
You guys ready for this? No.
Sir, you're being recorded saying that it's so dumb.
No, it's not. It's amazing.
Say that again. It's okay. Incredible. Sweet. All right. This is how we're going to remember it. Pyruvic acid combines with, you guys ready? You know it. Let's go. Coenzyme A to make acetyl-CoA. And it has to have the foot stamp at the end. All right? So it's coenzyme A to make acetyl-CoA. Ooh.
Sorry.
Let's see if we can do that together. Pyruvic acid combines with what? Coenzyme to make... Ooh, that's pretty bad. We're going to do that again. I'm going to do it first, and then you guys are going to do it with me. We're going to do it...
Form on the screen and you're telling us.
Okay, you want me to sit to form. All right, we'll sit to form. All right, so it's combining with coenzyme A to form acetyl-CoA. All right. I could picture it on the test. You guys are going to be taking the test and doing this. I can feel it coming already. All right, so let's do it all together. Pyruvic acid combines with what? Coenzyme to form acetyl-CoA. Oh, we got to get the foot stomp together. All right, let's do it one more time. Pyruvic acid combines with? Coenzyme to form acetyl-CoA. Woo, that was awesome. We should form like this biology stomping choir or something.
Okay.
Maybe not? Okay, we won't. That's a little, okay, I'm going a little far. Let's continue. Citric acid cycle. Okay, we've gone through the intermediate stage. We've combined with coenzyme A to form acetyl-CoA. And now we're going to the citric acid cycle. Citric acid cycle would be a series of reactions that break down the acetyl-CoA. There you go. It's breaking down acetyl-CoA to form, these are the things that we're forming now. ATP, we're forming NADH, and we're forming FADH2. Okay, so we're forming ATP. Do we want that? Yeah. Yes. We're forming NADH, and we do want that too, and we're going to see why in a little while. And we're forming FADH2. We're making, here are the specific details, 1 molecule of ATP. We are also making 3 molecules of NADH. And lastly, well, not lastly, but we're making also 1 molecule of FADH2. Carbon dioxide is a byproduct of the Krebs cycle. And this phase is very similar to the Calvin cycle in plants. And we've looked at that one. We're going to look at this one in some detail, and I'm going to show you the things that we need to remember. So these numbers, are they important for you to memorize? Yeah. Yes, they are. 1, 3, 1. 1 molecule of ATP. 3 molecules of NADH. 1 molecule of FADH2. 1 molecule of? Oh, we're starting over. I'm sorry, I didn't say that. 1 molecule of ATP. 3 molecules of NADH. 1 molecule of FADH2. And carbon dioxide is made, the reason we're not emphasizing it too much, this is a byproduct. Do we use carbon dioxide? No. No. Okay? When we Or maybe I shouldn't say that. All right, we don't use carbon dioxide, but plants use it. So it's a byproduct. When we breathe out, it comes out and all that good stuff. That's citric acid cycle. So we have here, you can see some of the things that we spoke about in the previous slide. Here it's using
coenzyme A.
no. What is this? Acetyl-CoA. All right, so we have acetyl-CoA coming in. That's important. So you might want to circle that, highlight it, put a smiley face next to it or something. Acetyl-CoA. Okay, important. Thank you for the foot stamp. Over here, important. We're making one NADH. Here we have another NADH. And here we have another NADH, which makes sense because we said we're making 3 molecules of NADH. Another important part here is we're showing that we're making one FADH2, and here's where we're making it. We're making 1 molecule of ATP, and you can see here and there that we have some carbon dioxide that is produced as a byproduct. Are we good so far? Yes, sweet. Then let's move on. So we've looked at glycolysis. We looked at the citric acid cycle. What do we have to look at next? Electron transport chain, or for the lazy people like me, ETC. Oh wait, no, not like me. I'm sorry. I didn't mean to say that out loud. Okay, electron transport chain. This is similar to what we've looked at before. In plants, it's a series of proteins. However, it's embedded in the mitochondrial membrane, and it's similar in idea to plant cells. But basically what we said, remember we said that the NADH, when it's making NADH, it's taking up an electron. FADH2 also takes an electron, and those carry the electrons to the electron transport chain. So we make the FADH2, we make the NADH, they take the electrons that they got, and they move it along to the electron transport chain. Are we good so far? Okay, sweet. They carry the electrons to the electron transport chain. Those electrons go down the chain, and it goes to the final electron acceptor, and that is oxygen. So I can almost guarantee you there's going to be a question on the test asking what is the final electron acceptor in the electron transport chain in cellular respiration, and you're going to tell me the final electron acceptor is oxygen. This is on the slide that deals with electron transport chain. I think actually in your handout it says something a little different. I think it says it's the final receptor of oxygen electrons. I like this better. It's the final electron acceptor because for whatever reason. Okay, so oxygen is the final electron acceptor. Oxygen then binds with H. What is H? Hydrogen to make H2O, and H2O is?
Agua.
Water, agua. Sweet. The electron transport chain, and this is very important. Electron transport chain. You remember before we were looking at how many ATP produced in glycolysis? 4 overall, and then 2 net. How many were produced in the Calvin cycle?
6.
Whoa. Where did we get that? Not 6, but 1. But in the electron transport chain, we're getting 36 molecules of ATP. Is that a lot?
Yeah.
Relatively speaking, that's a whole lot. So this is where we get most of the ATP for our body. Okay? Here we have an illustration of the electron transport chain. We have a series of proteins in the membrane of the mitochondria. We have NADH and FADH2. What do they bring to the electron transport chains? What are they bringing? It's the electron transport chain. What are they bringing? They're bringing the electron. Okay, the electrons. They bring those electrons to the electron transport chain. Those electrons go down the electron transport chain. What's the final electron acceptor? Oxygen. So it combines with oxygen to make, we're happy because we have H2O. Sweet.
But
Now,
when it went down
in
those
some
series
cases,
of proteins,
there's no oxygen
what did it release?
available.
Energy, and that
Okay,
energy is used to
and
make
in this,
ATP.
in these
ATP,
cases, we
how
go
many
through a process
molecules
of
ATP
ferment. We have a process of fermentation. Fermentation is a process that follows glycolysis when oxygen is not present. Okay, so if there's no oxygen present, we can go through the electron transport chain because remember the oxygen was the final electron acceptor. We can't go through that process, so we need an alternative process or an alternate process, and fermentation is a good one. Question?
Is this part of our lab next time?
Excuse me?
Is this going to be part of our lab?
This is going to be part of our lab next time. No, we're not going to be drinking any alcohol or anything.
We're making it though.
We actually will be you're excited?
I was kidding.
I'm just joking. All right, let's continue. But let's look at are we with me still?
Yes.
All right, sweet. We have two types of fermentation. First type of fermentation would be lactic acid fermentation. And the second type of fermentation would be alcoholic fermentation. And we're going to look at those two processes right now. Let's look at lactic acid fermentation first. Okay, here we have a guy. He's in the gym. He's working out. He's pushing some weight. Get it from the person next to you or something. He's in the gym. He's working out hard. He's bench pressing, for example. How much is he lifting? Give me a value.
500 pounds.
Whoa.
200.
500 pounds. Who is this beast of a guy?
No, that's like 180. That's like
Okay, I like 200. We're gonna go with 200 pounds. Okay, so he's lifting 200 pounds. He's on the bench and he's pushing. He does it 15 times. Second time he does it 10 times. Third time he does it 8 times. I don't know. I'm just calling out random numbers.
Okay.
Does he get tired?
Yes.
Okay, sweet. He gets tired. Does his muscle
No, I don't think so.
You don't think blah 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 blah?
No.
I think I need some water. My throat is kind of dry. You do too? From all the talking? Oh, okay.
Listening.
All right, let's continue. All right, so he's in the gym. He's working out hard. His muscles are using oxygen. Okay, so we have oxygen being sent to the muscles. Eventually, however, oxygen in the muscles run out, unfortunately. Okay, under anaerobic, what does anaerobic mean?
Without air.
Without oxygen. Okay, so under these conditions, the Krebs cycle and the electron transport chain cannot happen. They actually stop working. There's no oxygen to drive the process, so they stop working. In those cases, what happens is the two molecules of pyruvate, that's another name for pyruvic acid, those two molecules use NADH to form two molecules of lactic acid. Okay, why do we care about that happening? Well, the reason for that is NAD plus is then released so that it can continue to be used in the process of glycolysis. Why do we want glycolysis to continue happening? What do you guys think that would be? Okay, you can continue the glycolysis, but what do you get from glycolysis that's so important?
Oxygen.
No, you don't get oxygen. ATP. How many ATP do you make in glycolysis?
No, one.
36.
In glycolysis? Two is the net gain. Overall, you're making 4, and you're getting a net gain of 2. Is 2 better than none?
Yeah.
2 is better than none, so it does give you some energy so that you can continue using your muscles and continue lifting weights. Now, let me ask you this question. Let's say this is his first day in the gym. And why are you laughing at the guy? He went to Florida for Christmas break, and he's eating and...
Yeah.
All right, so let's say this is his first day back in the gym. What will happen tomorrow morning?
He would wake up.
He'll be like,
When he wakes up, he's going to be very
Stiff.
sore and stiff. His muscles might be burning and all that good stuff.
Get
Why?
out of bed and go to chapel.
Why would his muscles burn?
Because he ripped them.
Okay, that's part of it. Part of it has to do with the muscles actually tearing. But what do you think another part of it has to do with? This lactic acid build up in your muscles. Does lactic acid build up in your muscles, and do you think that's going to burn?
Yeah.
Yes, that's also going to contribute to the burning effect. It is an acid, and that does affect it. All right, so this does yield ATP, which is a good thing. Not as much as if everything were to happen with oxygen, but it yields ATP that can be used for energy. All right, let's go on to the next type of fermentation, which would be alcoholic fermentation. And that's exactly what we're going to look at now. Why are we looking at bread? Well, alcoholic fermentation is used by yeast cells, and we're going to look at this in the lab that we're doing on Friday. This is also when there's no oxygen available, and this produces carbon dioxide and ethyl alcohol. It yields 2 ATP. Is that a good thing?
Yes.
Yes. 2 is not as good as 36, but it's still ATP. It's still energy that we can use when there's no oxygen present or under anaerobic conditions. Now, how does that relate to bread? Do you use yeast in bread? No. Why? You cause to cause the bread to rise. How does it cause the bread to rise?
Using the ATP to puff it up.
Using the CO2.
What is CO2? Is that a liquid or a gas?
Yes.
Okay, so the gas would cause little air bubbles and so on, and that basically causes the bread to spread apart and basically rise. All right, so that's how the process of alcoholic fermentation works, and that is why we add yeast to bread so that the carbon dioxide can cause those bubbles, causing everything to expand. The bread is less dense, it's more fluffy, and we like that. Question?
Would the ethyl alcohol also be in the bread then?
It would, except usually when you're baking, is it warm or is it hot or cold? Okay, and alcohol evaporates very quick under higher temperatures. Okay, so when you're eating the bread, you don't have to worry about getting drunk or anything of that sort. All right, so in review, we've answered the question, what is cellular respiration? Process by which we make energy. We also spoke about the 3 stages of cellular respiration. And lastly, we looked at fermentation and how that happens. That's it for chapter 9. Remember, next class period, we're going to do a lab that has to do with fermentation. We're going to be using yeast. And class on Tuesday, we're going to have a review session. On Thursday, we're going to have a test. You guys look like you're excited to leave. Unfortunately, you're not leaving as yet. I'm giving you your homework assignment, and you can start working on that right now. So you might want to take your books back out and get excited.